Hi students, how are you? I hope everybody one can observe that previous lessons and previous the topics and now we are going to start that another topic very important in that particular NEET exam and that is the second chapter in second PUC, that is sexual reproduction in flowering plants. Sexual reproduction in flowering plants. Here, name itself present. Last class also, I told very clearly, there are different phases are present in the life cycle of the organisms, either plants or animals, especially in plants, juvenile phase, reproductive phase, sensing phase, and death and all. But when you compare to the animals, the plants has more survivability because of rapidly one can grow, because of meristematic cells one can be present and all. Then one among these one, in this the second stage of that particular plant, reproductive stage. Reproductive stage, already we said, what is the major indication of reproductive stage? Flowering of the plants in angiosperms. It is the indication of these particular flowering plants and all. In this reproductive stage, ready to produce the gametes and fusion of the gametes, formation of the embryo, then seed. All these process one can going on and on. The coming to normal our topic and all, here what are the points or what are the subtopics which one can be present? The subtopics are present. So what is the reproductive part present in that particular part and all? The reproductive part of the plant, that is the flower. So the first subtopic one can discuss the structure of a, what is the structure of a flower. Then second subtopic which one can discuss about that pre-fertilization events. Pre-fertilization events nothing but the formation of the gametes as well as that particular arrangement of these things, everything will be present under before fertilization under. Then fertilization, especially angiosperms, the special characteristic features of fertilization, that is nothing but double fertilization. Double fertilization, that one can be considered. Then post-fertilization. Then coming to the next one, post-fertilization, that means after completion of this one, then it will be formed, that embryo development, then endosperm, some of these particular plants, which one can be persist, which one can, it will be persistent. Some other plants, this endosperm, which one can be absent and all. That is depends upon type of the plants it will be present and all. Then after that particular post-fertilization included, they must be given that apomixis also. Apomixis. So here the structure of the flower, that pre-fertilization events, then fertilization, nothing but double fertilization, then post-fertilization as well as apomixis, this one. Then coming to the first one, related to parts of a flower. Already in lower classes, everybody one can be studied, but related to in competitive point, what are the major parts present in a flower? There are majorly it will be arranged into the different whorls and all. That is 4 whorls are present. What is the outermost whorl? That is the calyx. The second whorl, corolla. The third whorl, androecium. The fourth whorl, gynoecium. In these particular 4 whorls, these are the minimum basic things which one can be needed before discussing all these points and all. Then in this particular one, what are the accessory parts? Accessory parts are calyx as well as corolla. Then similarly, what are the necessary for reproduction? Necessary parts in that particular flower we called as, that is androecium as well as gynoecium. Andrecium is the male part, gynoecium is the female part. Then coming to in this that opening of the flower from floral bud, that process opening of the flower in that floral bud, that is we called as anthesis. Opening of a flower in that flower bud and at the time of maturation, that process we called as anthesis and all. In this one, that particular calyx, corolla, andrecium, gynoecium, but whereas in monocot plants, there is no calyx, corolla, but instead of segregation of calyx, corolla, it will be fused and formation of one particular combined part, that is we called as perianth and all. Then many myriads of flowers, myriads means very various numbers of flowers in our surrounding, that lily as well as rose as well as jasmine, such type of many different types of flowers one can be observable. But coming to in this competitive one, in this particular structure of the flower, that they may chance to ask what one can blooming, that formation of from floral bud, that is we call as anthesis. What are the necessary parts? Andrecium and gynoecium. The two parts very important. Then coming to the second one, the pre-fertilization. Then in pre-fertilization, that is we call as that sporogenesis. One can study about sporogenesis. Sporogenesis. Before fertilization, one can discuss about sporogenesis. In this sporogenesis, microsporogenesis, megasporogenesis or macrosporogenesis, that is micro as well as macro. Microsporogenesis related to the male, the formation of the gametes and all. Then macro related to the female. Macrosporogenesis as well as macro and as well as micro related to the male, macro related to the female. To avoid this one, very simply, you remember like this is the stamens, or this is we called as pistil. Stamens as well as pistil. As well as pistil, which one can be presented. Then coming to the first one, how in that pre-fertilization, microsporangium as well as and formation of the pollen grains and all. Microsporangium. So very simple, you must remember the micro in that sense related to the male part and all. Macro or mega related to the female part. So no need to say repeatedly. Now only you must listen very carefully. Micro, whenever I use it, that related to the male, macro related to the female. In that microsporangium, and which one can production of the microspores and all. From these one can produce the microspores. From where, which part of this one? Suppose in a flower there is in that male part it will be present, one stamen is present. The stamen, there is a male reproductive part and all. In andrecium, group of stamens are present. Stamen we called as microsporophyll. Stamen, this is stamen. In stamen we called as microsporophyll. That matured stamen. In that stamen, what are the parts are present? One is that filament part. Then another one is the anther part. This is the connective. This is very important. Wherever from filament which connected to the anther, that particular area we call as connective. Normally, many people one can remember only anther as well as filament only. No. Connective also one among the part will be represented. But here, very important part for the production of the male gametes and all, that is the anther and all. What is the another name of anther and all? Male sporophyll. This is male sporophyll. Don't confusion microsporophyll stamen, male sporophyll anther. In that anther, it will be present, the two lobes are present. Two lobes are present. That is, if it is present two, that is we called as dithecus. If it is present one, that is we called as monothecus. Then in exam point of view, many times they ask regarding related to in that dithecus, that how many number of pollen sacs, which one can be present inside these particular aspect syndrome. In that pollen sacs, suppose in that dithecus, in that dithecus, 4 pollen sacs are present. Dithecus, it will be present 4 pollen sacs. That means if you observe in a TS of anther, one can be observable. TS of anther, this is the connective part. Connective part, these are the pollen sacs. This is we called as stomy. So, in the TS of anther, so these particular parts, name of the labeling, that is most important. Many multiple times in exam point of view they ask random. Then the outer covering, normally it will be present. It will be present that epidermis. Epidermis layer, already it will be covering. Already it will be present. Then lower endodermis, that means the hypodermal layer. Hypodermal means below the epidermis. Hypodermal layer, one specific cells, it will be present. That is we called as archisporial cells. Hypodermal layer, a specific cells are present. That is archisporial cell. Archisporial cells are present below epidermis, that is hypodermal and hypodermal layer. In that archisporial cells, which one can divided mitotically, and 2 cells are formed under that particular 2 cells, outer parietal cells, lower sporogenous cells under. In this outer parietal, then inner sporogenous cell. So below this one, there are 2 types of cells. It will be present like parietal as well as sporogenous. Parietal cell and sporogenous cells are present. Then in that parietal cells, especially in parietal cells further mitotic cell division, which one can differentiate different types of the layers, that which one can be formed periclinally. Periclinally, it will be undergoes the mitotic cell, that is very important here, either meiosis or mitotic. Here archisporal cell, periclinally mitotic cell division, and one can be formation of different layers of cells. What type of layers of cells normally one can form from archisporal cells? Archisporal cells, then outer, inner, then in this parietal cell. From parietal cell, one can be formed a single layer of endothecium, a single layer of endothecium, as well as middle layers, then tapetum. Middle layer as well as tapetum, that it will be present under. Then in this, that 3 particular layers are present. So parietal, endothecium, single layer, this is the fibrous one, and dead cells, fibrous as well as dead cells, very very important, fibrous and dead, and a single layer and diploid condition. 2n, their chromosomal number. Then middle layer, minimum 2, maximum 4. 2 to 4, that particular layers, it will be present, middle layer. Then the innermost, that is the tapetal cells. So in exam point of view, many times they asked that related to, so which part of these cells, the tapetum cells, which one can be arises? Then they have given options, either endothelium or middle layer or tapetum or epidermis or many other options one can give. Then you must write the option, that is the parietal cells. So from parietal cell, tapetum layer, it will be formed. Among these one, it acts as a very crucial role and all. Then sporogenous cells, one among the sporogenous cells act as a pollen mother cell and all. Sporogenous cells, one can form microspores. In this, one among these one act as pollen mother cell, again a diploid condition. Through meiosis cell division, pollen mother cell, which one can undergo, then one can form microspores. Microspores nothing but pollen grains or male gametophyte. Microspore or pollen grain or male gametophyte, one can be formed from sporogenous cell. One among these sporogenous cells, during maturation, it acts as diploidic condition, which one can undergo meiotic cell division, and one can form haploid, that to particular pollen grains and all. That means that is the microspores or male gametophyte or microspores are present and all. That is, so all these, the particular outer epidermis, here once again we can write outer epidermis, this is outer epidermis, then this is the second layer, endothelium. Then for understanding purpose one can write like this manner, minimum 2, maximum 4 it will be present, that is the middle layers are present. Innermost one, this is the tapetum present. Inside this one, that one among this one, that is sporogenous cells, which one can undergo, and formation of the male, that microspores, it will be formed under. Then this is the dead cells, endothelium. So especially epidermis protection under. Then what is the function of that particular endothelium? Helpful dehiscence of the pollen grains, dehiscence of the microspores. Dehiscence, that means dispersal after formation, which one can rupturing, which one can separation, which one can dispersal of this one. Then related to middle layers also very short-lived and it is also present minimum 2 and maximum 4 layers it will be present under. So 2 to 4 layers of middle layer it will be present. So all are diploid, endothelium diploid, epidermis diploid condition, middle layers diploid condition, but tapetum polyploid. But another very important, who is the scientist who must be explained related to tapetum cells? That is Ubish, one botanist scientist, one can explain it. The name of that scientist, Ubish scientist, explained about what is the function, how many types, everything one can be explained. Then what is the important function of tapetum cells? There are the important function which one can nourishes of these microspore, further formation of microspores and all, as well as which one can nourishes, secretion and absorption. Very most important, once again remember, nourishes, secretion, absorption. These 3 particular function one can involving in the tapetum cells. Ubish scientist, one can explain about tapetum. According to you which one can be present? There are two types of tapetum cells are present. One is the amoeboid cells, another one is the glandular types of cells. Amoeboid cells, otherwise we call as invasive cells, or we call as periplasmodium cells. Then, what is the important function of amoeboid cells? Here only, that's why I have written different shapes and all. One is amoeboid, another one is the glandular. But amoeboid cells helpful for formation of exine, the outermost layer of the microspore. Then another type of cells related to glandular cells. Glandular cells are parietal cells. What another name of glandular cells? Parietal cells. These are, one can secrete different types of the chemicals like hormones, like particular nutrients. It is the persistent. What type of hormone one can secrete? IAA, indole 3-acetic acid. Apart from this one, it is also secretes one type of the granules. Lipid granules are secrets. What is the name of that lipid granules? Ubish lipid granules are present. These ubish lipid granules, it consists of sporopollenin. That is most important. Sporopollenin. That sporopollenin, it will be present in that. So, in these tapetum cells, whatever one can say, like amoeboid as well as glandular. In this one, amoeboid, one can be secrets, one can be, which one can be present, and formation of the exine in that pollen grain center, which are outermost layer under. Then glandular, which one can secrets that hormone as well as nutrients as well as some among these ubish granules. Ubish granules only, it will be contain sporopollenin. Sporopollenin. So that is, so the shortest living, that is we call as this one, middle layer of the cell center. So, and most important, endothelium, decision center, important function, that is tapetum under, which one can act as important role. Then coming to sporogenous cells, one among these sporogenous act as this one, pollen mother cell. Then one can undergo meiotic cell division. So once again, all are that ploidy, very very important. What is the ploidy of epidermis, endothelium? So all already we have written. Once again you observe very carefully. Tapetum only polyploid. Why it is polyploidy? Endomitosis. Because of endomitosis, minimum 3n number, which one can present. In this one, what type of cells in that tapetum, which one can be persistent, which one can be, it will be present maximum period of time. In this one, parietal cells or glandular cells. Then the first type of amoeboid cells, immediately which one can enter towards that particular sporogenous under, and it will be degenerate. Then before descends, that particular type of glandular cells also degenerate. So until descends, all it will be present. So finally the tapetum nourishes absorption as well as these, all these particular important role one can be takes place. That scientist one can say, that is we call as division. Then one among the pollen mother cell undergoes meiotic cell division, then it will be forms into the microspore center. What is the gamete? What is the ploidy of this microspore? Only haploid. Always it is the haploid condition, microspore center. Then normally, how many? What is the condition which one can form into the haploid and all? That is the pollen. It will be present into the pollen tetrad. It forms pollen tetrad. Normally, minimum 4 pollen which one can be present. Apart from this one, pollen mother cell, due to meiotic cell division, which undergoes and formation of four cells one can be formed under. Then 4 cells it will be present. Among this one pollen mother cell After meiotic cell division, pollen mother cell that is normally diploid in condition, that is undergoes meiosis cell division, then it forms into the pollen tetrad. That means 4 haploid microspores are formed under. 4 haploid microspores, that means pollen grains are present. In this particular pollen grains, in that tetrad, that means minimum 4 cells which one can be present. 4 cells which one can be present. In that pollen tetrad, that 4 cells. That means pollen grains. Then, what we call the study of the pollen grains? Palynology. Study of pollen grains. How many layers are present each and every pollen grains? Outer exine, inner intine it will be present. Then outer exine, in that outer exine, it is very tough or very withstand, very resistant. Then with the help of that particular chemical, one can store, one can preserve long period of time. So normally one can preserve minus 196 degrees liquid nitrogenous solution, one can be preserved for a long period of time. Then what is the important role? Why one can preserve these pollen grains that much long period of time? Because of sporopollenin.